Welcome to Introduction to Diffusion, presented by PermeGear. So what is diffusion? At its core, this is what diffusion is. You have an area of high concentration which spreads out into new areas at lower concentration. This applies to nearly everything, liquids, gases, and solids, though solids tend not to move a lot on their own. Transdermal and topical products are not new, far from it. Many basic products date back hundreds, in some cases over 1,000 years. For example, a cold cream was created before 200 AD that has composition similar to current products. A variety of products fall under this umbrella. Cream, gels, and ointments are quite obvious, but patches are counted as well. There are even newer technologies like iontophoresis and microneedles which fall into this category. Diffusion testing can be used for a variety of applications, but we'll focus heavily on pharmaceutical applications for this presentation. This includes topical and transdermal products like creams, gels, ointments, and patches. For nutraceuticals, they have values in a few different ways, primarily to evaluate marketing information where the product is applied to the skin, and then the skin is evaluated afterwards to determine the effects. This can be considered a strong cost-saving measure as it reduces the need for clinical testing. In vitro studies, in addition to other information, can help with any FDA or other regulatory agency reviews of the products. Cosmetics fall into similar categories as nutraceuticals, but can provide additional data to prove ingredients do not affect the skin. Sunscreens are a good example of this, as they need to be tested to ensure ingredients are not entering into the deeper layers of the skin. This was evaluated extensively by the FDA only a few years ago. Another category applies to those who manufacture membrane barriers. In this case, the diffusion coefficient of the membrane is evaluated often with a saline or similar solution. Horizontal or side-by-side diffusion cells are more often used for this than Franz cells. Diffusion testing as it pertains to pharmaceutical products is generally separated into two broad categories. The first is permeation testing. This is done to evaluate formulations and get an idea how they perform when used by a patient. It's meant to be biorelevant. The receptor media is often phosphate buffer or a saline solution, and the membranes are usually natural, being excised skin or something meant to behave like skin. This unfortunately means the amount in the sample may be in picograms per milliliter, so a sufficiently sensitive analytical method is a must. There can also be other processes performed after the test to determine how much of the API made it into and stayed in the skin, or even specific layers of the skin. Release testing is more focused on comparison of two products. This can be a comparison between a generic and an innovator product, or testing to see if a certain ingredient or process change made a significant difference with the finished product. This test is not biorelevant and is run with synthetic membranes and alcoholic mixtures for receptor media. The idea of this test is to find out what is the unhindered release rate of the product. Typically, a test product and a reference product are compared, and if the results match within a 90% confidence interval, the products are considered the same. Permeation testing fills a particular role in the pharma industry. It's currently the only in vitro test which can give a good idea of product performance without the need for costly clinical trials. This allows for screening formulation and saves a lot of time and money on R&D. In some cases, when a manufacturer is looking to produce a generic product, this may eliminate the need for clinical testing entirely. As a potential QC test, it fails though, but that's not necessarily the fault of the test itself, but rather the skin used as membranes. Even on the same person, skin is highly variable. Compare the skin on your forearm to the skin on the bottom of your foot. There's also variations across the body for skin that feels the same but performs very differently when it comes to topical products. This is exacerbated in testing, as different donors will almost certainly have different skin, but variations within the same donor can exist as well. This means the results for product performance may be highly variable unless dealing with a fairly large sample pool. Combine this with potential detectability and quantification challenges from samples in the picogram range, and it's easy to see why this test is exclusively used by R&D. IVRT is an entirely different type of test than the permeation test, and has a very different objective. Primarily, it is used to compare the release rate between two variations on what is essentially the same product. This can be generic versus innovator, or on the same product before and after a change in an ingredient or process is made. A difference in the result does not guarantee there is a difference in bioavailability. The nice thing about this test is the results tend to be very consistent. The membranes are synthetic, often the same material found in syringe filters, so there is none of the inherent variability found in using skin as a membrane. Additionally, the sample concentrations are relatively high compared to IVPT due to the media being focused around attaining excellent sink conditions, often to the point of including inorganic components such as methanol or ethanol in the receptor media. The downside is there is absolutely no in vivo in vitro correlation, so this test really only has value when you're comparing one product to another. It can tell you if something's changed, but it can't tell you if that change is significant, so the possibility is there that it could cause a need for an investigation only to find any issues are of no consequence. There are a number of different ways to perform diffusion testing. The Franz cell or vertical diffusion cell is the most common. It covers a variety of different types of testing and has a very well-established background in the literature. It can be customized by manufacturers to have different surface areas for the orifice as well as different receptor volumes, making it incredibly versatile. It's a very good choice for general purpose testing where the method and results will be reviewed by a regulatory agency. There are a lot of variations on the design between manufacturers, and the right one really comes down to the requirements of your study. It is recognized as suitable for both permeation or release testing. The inline or flow-through diffusion cell has a stronger focus on permeation testing. The media flows through the cell the same way the blood moves under the skin in a person. This constantly removes the drugs, so there is no buildup, which may slow down the rate of diffusion. This is a great choice for formulation development, as it can be a big help with poorly soluble products, as you can easily adjust sink conditions by adjusting the flow rate. These are less common overall than the Franz cells, but still widely recognized. Side-by-side or horizontal diffusion cells are a more specialized instrument. They hold a membrane between each half. One half is the donor chamber, while the other half is the receptor chamber. This is not often used for testing pharmaceutical products and has a higher focus on determining membrane diffusion constants, though for certain type of tests there may be pharmaceutical applications. For this, it is required that both chambers be completely filled with liquid as compared to Franz and inline cells, which will function fine if the donor chamber is only partially filled. Diffusion cells of any type come down to three basic components: a donor chamber where the high concentration of the material resides, a receptor chamber where the material will diffuse to, that is kept of uniform concentration, and a membrane that separates the two. Common features, though not shared across all designs, are stirring of the receptor chamber and temperature control. The Franz diffusion cell is probably the most recognized and most versatile of diffusion testing apparatus. The basic construction is what appears in the diagram here. It includes a donor chamber and receptor chamber. The membrane is placed between the two chambers. This cell has a water jacket which is typically hooked up to a circulating water bath to maintain the temperature. There is a sampling arm on the side. This arm is designed for a needle or pipette to go in and pull a sample from the center of the receptor chamber. Other models are designed for the entire contents of the receptor chamber to be poured out, which can have some advantages during analytical steps. Unlike some of the other apparatus we'll discuss, Franz cells are widely used for both IVRT and IVPT testing. For groups just starting diffusion testing, these are generally the best apparatus as they can handle just about everything. Orifice diameter and primarily the receptor chamber volume can be customized. There are limits, however. For example, you can't have a 30 mm orifice diameter with a 1 milliliter volume, but within reason there is a lot of flexibility. The joints can also be customized. Flat ground glass joints are primarily used as they seal well on a variety of materials. Spherical joints are available for corneal research, and there are also O-ring joints which can be suitable for very thin membranes or testing of gases. The donor chamber types can also be modified. In some cases it's as simple as adding caps, but there can also be ports put in for flowing gases through the chamber or additional ports for added probes. Generally, the horizontal diffusion cells are very similar in construction to the vertical diffusion cells. They feature many of the same options. The key differences are orientation, with the membrane held vertically rather than horizontally, and with the donor chamber. Typically, the donor chamber is the same volume as the receptor chamber. Both chambers have stir bars present to add circulation. Additionally, both chambers have sampling ports which can be used for filling the cells and withdrawing samples. Unlike vertical diffusion cells, which are heavily focused on transdermal applications, the side-by-side diffusion cells are typically used for determining diffusion of liquids or for determining how materials may diffuse through certain membranes. For example, how a specific salt or drug may pass through dialysis tubing. Inline or flow-through diffusion cells are a completely different design but have been around a long time. Unlike the Franz and side-by-side cells where the media stays in place, the inline cells constantly have media entering and leaving the receptor chamber in a continuous flow. The flow rate is typically slow at only 8 to 16 microliters per minute. However, because it is constantly being moved out of the cell, there is not the same level of buildup of the active ingredient that one would see with Franz diffusion cells, making sink conditions much easier to maintain. There is still temperature control, but in the case of the inline cells, this is from a heated holder rather than water jackets. The inline cells have a focus on transdermal and topical products, much like the Franz cells. However, they are more specialized and tend to be used almost exclusively for permeation studies. IVRTs are possible, but it's rare to see any published data. The inline cells do have another advantage over the Franz cells in that they are very easy to automate. This allows for automatic sample collection, which means lab staff don't need to stay overnight to withdraw samples in time. Over the past few decades, a number of documents have been created to help guide manufacturers to good practices for creation of and testing of topical and transdermal products. The first was the SUPAC SS, which was created in 1997, which defined how to test a reference product against a test product, and defined the use of the Mann-Whitney rank test as a means to evaluate the sameness of the products. This method is still used for evaluating in vitro release tests today. USP 1724 was created to further define this, as well as provide guidance on the types of instruments typically used for in vitro release testing. A revised version of USP 1724 was released in December of 2023, which includes not only updated information for IVRT, but IVPT as well. The FDA guidance on acyclovir cream provided a significant amount of detail on in vitro testing to the point it was often reviewed by groups that do not manufacture acyclovir, as the information can easily be applied to other products. The FDA also released 3 guidances on semi-solid products in October of 2022 covering basic quality tests, IVRT, and IVPT. In 2018, the EMA released a guidance on the quality and equivalence of topical products, which covers a wide array of topics, but has 2 appendices which cover IVRT and IVPT specifically. And the FDA released a more general purpose guidance on transdermal and topical delivery systems, product development, and quality considerations for patches. Thank you for viewing this video. If you have any questions or would like to know more, please visit us on our website at PermeGear.com or reach out to your local PermeGear distributor.